Hi there, everyone. We are so happy to have you, whether you are joining us here today or will be watching from our Cure GABA-A YouTube channel. We ask that you please mute your mics if you have not done so already, and include any questions you might have in the chat box below. We'll have time to answer questions at the end of the presentation. My name is Monica Joanna Elnakabe, and I'm the founder and CEO of Cure GABA-A, a nonprofit organization with the sole focus to develop potential therapies for GABA-A variants. By partnering with world-renowned researchers in GABA-A and the Organization of Patients for Clinical Trials. Today, we welcome Dr. Henry Lee of Boston Children's Hospital, who is a molecular neurobiologist specializing in understanding how molecular processes influence neuronal inhibition, brain plasticity, and the recovery after brain trauma. His research, which integrates molecular biology and electrophysiology techniques, has led to significant discoveries, including the role of KCC2 in controlling GABAergic inhibition. I'm handing it off to you, Dr. Lee. Please explain to us the understanding of GABAergic signaling.
Well, thank you, Monica. It's a pleasure to be here and talk about some of my work at Boston Children's. And also, as you described, some of the work related to KCC2, which actually is my PhD work. So let me just share my screen. All right, so good afternoon, everyone. So my name is Henry Lee. I'm a senior scientist in the FM Kirby Neurobiology Center at Boston Children's Hospital. I also have another position in the Boston Children's Hospital as a preclinical science program manager in the RSC Translational Neuroscience Center. So today I'm going to talk about some of my PhD work and the ongoing work at Boston Children's, and circling around a theme of understanding GABAergic signaling. So my understanding is that this is for a Cure GABA-A variant community, and my talk is more tailored to a more basic understanding, and I try to strip it down to a more fundamental understanding of this of what I have done. So just a little disclosure before my talk. I'm co-founder of Galiber Neuroscience. It's a startup focusing on gene therapy, and I'm also a co-inventor of the patent gene therapy in succinic semialdehyde dehydrogenase deficiency, SSADHD. And the end of the talk, I will talk a little bit about this specific disorder. The content of my talk today I will break it down into three subsections. The first part, I will talk about what is GABA and what does it do? What does it signal in our brain? And the second part, I will talk about my PhD work focusing on a molecule called KCC2. How does that modify GABAergic signaling? And at the end, I will talk about a specific genetic disorder due to failed GABA recycling. So what is GABA? What does it do to the brain? So for a community in the Cure GABA-A, GABA should not be that sort of stranger, but what is GABA actually? So it's a short form of gamma aminobutyric acid. So GABA is actually a little bit of history of that. It's actually first described by Eugene Roberts in the 1950s. And it has been found to be produced and accumulate in the brain with a high amount. And but it's not until 1957 when GABA is now understood to be a major inhibitory neurotransmitter. So what does it mean by a neurotransmitter? So what does it do? How does it work? So GABA is a ligand or a receptor in the brain that we call GABA receptor. So this cartoon shows you a graphical representation of this GABA-A receptor. The basic structure of GABA-A receptor is made of 5 subunits. So what we call a heteropentamer structure. So basically, it's 5 subunits that bound together. And you can see this cartoon showing that it is floating in this membrane location. So these 5 subunits, a typical form will be consisting of 2 alpha subunit, 2 beta subunit, and 1 gamma subunit. So nature is very interesting. These units will find their way and then they form this functional receptor. And So far, we have known that there are 19 subunits, so including 6 isoforms of alpha, 3 isoforms of beta, 3 isoforms of gamma, 3 isoforms of rho, delta, epsilon, theta, and pi. So these heteropentamer is the basic structure of forming a GABA-A receptor. So this is important to know that what does a receptor do in this case? There are positions where we have known that the GABA can bind. For example, GABA bind to this interface between the alpha and beta subunits. And benzodiazepine, a drug that potentiates GABA receptor function, binds to a different location in the GABA receptor. So what does it do when this ligand or this modulator bind to the receptor? What it does is that it opens the channel, allow chloride ion to move. So this becomes very important later in my talk. And so just for now, we want to know that GABA receptor is a chloride channel. And when GABA binds, it opens the channel and let chloride move across this membrane. So just a little bit basic neuroscience to talk about what is the transmission. How does neuron communicate with each other? So this is a cartoon showing you a neuron from what we call a presynaptic neuron and a postsynaptic neuron. The synapse is the site between two neurons where they communicate. So in this case, there is a nerve impulse signaled by this graph here, and it will travel along the neuron and go to this portion. And what it does is that there is a switch of the polarity across this neuronal membrane, as shown by this cartoon. And one by one, this impulse will be communicated and then transferred to the end, the end of this neuron. And what it does is that there will be a release of the neurotransmitter across this synaptic gap and then act on the next neuron for this relay. So there are two consequences of this synaptic transmission. One is that when the neurotransmitter act on this receptor, changes in the postsynaptic neuron in terms of their polarity can be very similar to the presynaptic neuron. You see that is a action potential. So in that case, if there is a flip of the of polarization, then now it becomes a depolarized neuron, and the action potential can carry on further. The other alternative is that there could be a strengthening of the negative charge. In this case, there will be a hyperpolarization, and that will actually inhibit the neurons from firing. So this is the inhibition. So why GABA becomes inhibitory is because of this chloride channel. When this open, the chloride move in, resulting in a inward chloride movement. And that will lead to a hyperpolarization event, and the postsynaptic neuron becomes more difficult to fire. So this basically is the basics of how GABA becomes inhibitory. This cartoon also shows you there are some relevant transporter that is maintaining the chloride. So the chloride becomes a key for why GABA-A receptor is inhibitory, because the chloride movement is from outside to inside, so hyperpolarizing this neuron and then to make it more difficult to fire. But this is not the whole picture. This is actually happening in the mature neuron. And now we know that in immature neuron, there is actually an opposite scenario where chloride is actually very high inside the neuron. And in that case, GABA-A receptor actually becomes less hyperpolarizing, or in some case, depolarizing. The reason is because chloride move out, so causing a flip of the or depolarizing event. So in this case, GABA becomes less inhibitory. And this is actually a natural phenomenon in a maturation of a neuron. So this is what we know. And for this part of the summary, well, now I'll just describe to you that GABA binds to the receptor. And in this case, GABA-A receptor is made of this heteropentamer structure, 5 subunits. And they are governing the chloride movement across this transmembrane location. And because of the chloride is naturally lower inside the neuron, there is a net inward movement of the chloride. And this inward chloride movement will lead to hyperpolarization and neuronal inhibition. But that being said, that means that GABAergic signaling, particularly through the GABA-A receptors, can be modified by this chloride balance. So this leads to the second part of my talk, is talking about how this KCC2 molecule modulate or modify this GABAergic signaling. So this is my PhD work, and under my mentor, Stephen Moss, now at Tufts University. And this is back in 20 years ago, when I started my PhD work in the Moss lab. At that time, we just know a little bit about how this GABA or how KCC2 is a chloride co-transporter, potassium chloride co-transporter that moves chloride out of the cell. So this, at that time, we know that it's important for maintaining a low chloride concentration inside the neuron, and that's why GABA becomes hyperpolarized. At that time, we don't know much about how KCC2 is regulated inside the neuron. What are the factors that might change KCC2? And it turns out to be very important. That time, Dr. Moss persuaded me to start this project, understanding how in the neuron, how there are molecular events that might change KCC2 function. So I just use my molecular cloning technique and first study this internal tail of the KCC2. You can consider this KCC2 as a string of beads that are, intercalate in this neuronal membrane. And there are N-terminal and C-terminal, these tails that are anchor for KCC2 on the membrane. At that time, we don't know much about how KCC2 is regulated, but from previous study in Dr. Moss' thesis, looking at the GABA receptor, we know about how protein phosphorylation might change the protein insertion into the membrane. So at that time, using some similar bioinformatics technique, we can actually guess that there might be some amino acid residue on the KCC2 tail that might be relevant for protein phosphorylation, and they might have a role in stabilize or destabilizing this KCC2 on the membrane. So using a in vitro technique, basically extracting this C-terminal tail of KCC2 and run it in the test tube, and then see how protein phosphorylation might be or might not be acting on this tail. I'll also do mutation of this. So first off, there are 3 residues that are potential candidates that might be phosphorylated in KCC2. And I mutate them one by one and then look at the amount of protein phosphorylation. And it turns out I hone it down to one residue called serine 940. And this mutation will render the phosphorylation reduction in the KCC2 molecule. So this becomes a major site at that time that we think that will be related for KCC2 phosphorylation. And it turns out this this is a radioactive isotope labeling experiment. So let me just explain a little bit here. Is that so we have a wild type, a normal tail of the KCC2, and we use a radioactive isotope to label them. And by labeling, you see this band, that means that the protein is phosphorylated. And I mutate these sites one by one. And by mutating it, you can see how the amount of this labeling might be affected. In this case, I don't see a change in this serine 728. I do see a very high reduction of this band. You can see reduction in the serine 940. And on the other hand, the third site, serine 1034. So these 2 sites seems to be not affected, but the serine 940 seems to be the key that can be related to the protein phosphorylation. Additional of the combination of different of these sites doesn't seem to change this lowering of phosphorylation. So suggesting that this serine 940 is a major site of protein phosphorylation for KCC2. So what happens to phosphorylation if we mutate that site? Then we test it in a neuronal setting. We use a chemical, PDBU, to activate a protein kinase, which is an enzyme for protein phosphorylation. And then we ask the question, if we activate this protein phosphorylation process in KCC2, what does it change to the cell surface? So we can use the biochemistry method to label or to understand the cell surface expression of KCC2. So this, again, this is a band that should show that there a in the normal KCC2 molecule, there is a cell surface presentation of KCC2. And if we activate with this chemical, PTBU, we can see the increase of this cell surface presentation, suggesting that activating PKC, activating protein phosphorylation will make more KCC2 on the cell surface. So you can actually see that trafficking is increased. However, when we mutate this serine 940 into a residue that is unable to do a phosphorylation, now you can see that PTBU lose its effect. So it does not increase anymore, suggesting that this mutated KCC2 is impaired. It cannot be inserted into the cell surface. So this has a functional consequence. So this is published in 2011. And what we hear, we're showing that using a glutamate, so this is another neurotransmitter that we know to affect the protein phosphorylation. So by challenging the neurons with this neurotransmitter, we can actually see a very quick, in terms of timing, you see that only 15 minutes of this challenging with this neurotransmitter is able to reduce the this phosphorylation of the serine 940 residue of KCC2. At the same time, we see a reduction of the total amount of KCC2. So not surprisingly, on the surface, KCC2 is also reduced by this treatment. So this is a molecular measure of how protein phosphorylation will affect the protein amount on the surface. But also, what about the function? What about its GABAergic function? So actually, the title already gave out this idea. So we actually measure the signal of this GABAergic response. And before the challenging, you see this downward movement. If you still remember, this becomes inhibition. So GABA normally is inhibition. But after this challenging of this glutamate neurotransmitter, we see a reverse of that. So a lot of GABA now becomes not inhibitory, but excitatory. So this is actually the first of the evidence showing that KCC2 function and link to the GABAergic signaling can be quickly modulated. So by just 15 minutes of the challenging of the neurotransmitter, you can actually modify or modulate the amount or the polarity of GABAergic current. So what happened to the mouse? So now we want to put it into the context of a maybe a potential mutation scenario. So we make use of a genetic method to mutate this serine 940 of the KCC2 in a mouse. And this is actually a electroencephalogram, EEG, of a mouse, basically measuring the brain wave, the pattern of an otherwise normal mouse in their when the animal is freely behaving in their arena in a home cage. And this is a recording up to 120 minutes, and you can actually see this arrow is the time when we challenge this animal with a chemical that will make them seize. So you can actually see that before the challenging, the animal is behaving normally, but then they start to have this more higher activity of the EEG, suggesting that they start to having the seizures. So you can see that activity becomes progressing and then becomes a more rhythmic and uncontrolled seizure event in here, eventually within 2 hours. And what happened to the mutated mouse? Without this serine 940 phosphorylation, you can actually, even at the baseline, you can see this activity is higher. But upon the challenging of this chemical that makes them seize, you can see the seizure becomes much more violent and much more earlier. And this is actually out of control at the point where this arrowhead signals the death of the animal. So the animal becomes much more susceptible to seizures without this able to phosphorylate of the QCC2. And it also cannot control the seizures to a point where it becomes mortality. So this is actually towards the end of my PhD work that where almost 6-7 years of my work resulting into a mouse that shows that this only one amino acid residue on KCC2 can change the GABAergic signaling and have a very meaningful functional effect. So now 20 years later, we know much more about how KCC2 might be related to seizure control. There are also mutations that has been documented that is related to human epilepsy. So, of course, KCC2 now becomes a very viable pharmacologic target for anti-epileptic seizures and anti-epileptic drug. So these becomes a very hot topic right now. It's a race for making drug that can activate KCC2. So to summarize this part, just quickly, is that KCC2 is a phosphorylated protein. So what does it do? Actually, it can change the stability of KCC2 on the neuronal cell surface. By doing so, we can modulate the amount of transmembrane chloride gradient. And in some situation, if we reduce KCC2 function, that will lead to a accumulation of chloride. And that time, GABAergic signaling becomes less inhibitory, or in some case, it could be depolarizing. So, KCC2 is a very hot topic for a therapeutic target for seizures. So the third part of my talk, I would like to change gear to a little bit to talk about the genetic disorder that related to GABA recycling. So I want to zoom out a little bit. What I just talked about is the GABA and acting on the GABA receptor, and because of the chloride gradient change and lead to inhibition. So what about the GABA that got released, and what is the life cycle of GABA in the brain? So it turns out there are other research that is studying how GABA can be recycled. And after release, there are at least some mechanisms for GABA recycling back to the neuron or the adjacent astrocytes, so other cell types that is important for the support of the brain function. There are known at least GABA transporter 1 and 3. They are in the neuron and autoastrocyte that are relevant for the reuptake of GABA. And also after reuptake, there are mechanisms to degrade GABA. So degradation of GABA is 2 steps. One is GABA transaminase, GABA-T, and then converted to succinic semialdehyde. And this SSA will be further degraded by SSADH, succinic semialdehyde dehydrogenase, into succinate. So this last part of my talk is focusing on this disorder where succinic semialdehyde dehydrogenase is impaired. So this is a condition called SSADH deficiency. This is a rare disorder and without cure. So this is the work, now I work in Dr. Rothenberg's lab in Boston Children's Hospital. And what is SSADH deficiency? It is an ultra-rare disorder. It's less than 500 documented cases as of this year. And the reason of this deficiency is because of an absence of SSADH activity. And this is an enzyme encoded by a gene called ALDH5A1. So it's 2 copies of this gene is mutated, and the patients do not have SSADH activity. And what does it do is to degrade GABA, as I just mentioned in the last slide. And this is a biochemical cascade of how GABA is degraded in neurons. And the first step, as I mentioned, is GABA transaminase, degraded to SSA, and further degraded to succinate. Without SSADH, the second step is missing. But what happens is that there will be a equilibrium to push this biochemical equation back to GABA. Or alternatively, there is a alternative pathway that will convert SSA to gamma hydroxybutyrate, GHB. So both GABA and GHB accumulate in the brain of a patient to a pathologic level. But interestingly, many of the patients have seizures. So this is a science aspect that we want to understand why accumulation of inhibitory neurotransmitter will lead to epilepsy, will lead to excitation. So this is something that becomes relevant when it comes to treatment. Right now, there is no treatment. The treatment options are very limited, and they are not effective, mostly because the treatments are not targeting the underlying cause, which is the absence of SSADH. How do you target the underlying cause? Gene replacement therapy will be one potential viable approach because we know precisely where the gene is having a problem, and we also know that where the gene is expressed. So a gene replacement therapy could be a potential cure, but so far it's not available, it's not developed yet. And that's where we want to understand better. In here, um, so we have some idea what gene therapy might be usable in this situation. But first, we also want to understand the science behind SSADH deficiency. So we think that in a normal situation, we have a balance of GABA and GABA receptors. In SSADH, because the degradation enzyme is missing, so we have accumulation of GABA. At the same time, the brain is compensating this accumulation, so they have reduced GABA receptor to try to find a balance. But for some reason, this balance becomes overcompensation, and there's a net reduction of inhibition. And that might be explaining why patients have seizures. And this actually is also, there are clinical evidence suggesting that in the SSADH patients, they have a reduction of availability of the GABA receptors. And but when it comes to gene therapy, we are mindful that the enzyme that we introduce into the brain will lead to a renormalization, meaning a reduction of GABA. So at the same time, the brain needs to be reverse this compensation of the downregulation of GABA receptor. So in other words, we want to have a reinsertion of the GABA receptor into the specific place for this balance to be restored. So is it true? So can we mimic that in the mouse? We want to, before we develop the gene therapy, we want to test it in a mouse model. So we want to, so what we did was to make the mouse that has a stop codon. So essentially is to make a switch to switch off the gene of the SSADH producing gene. So in this case, at the before manipulation, the mouse becomes a sick mouse because they don't have SSADH. They have accumulation of GABA. But we can use a molecular method to switch on the gene. And by doing so, then we restore the SSADH activity in the brain and in the whole animal. And we basically want to test whether this approach might be already useful to see the reverse or the rescue of the mice. So here is two videos to show that what happened before and after switch on the gene. So let me play the video. And as you can see here, this is the before treatment. This is a pair of animal. One is with the switch and is switched off. So that means SSADH. So you can see the movement and also seizures later on in this video. The other animal is a littermate, basically same, not the same animal because this does not have a the switch to switch off the gene. So so you can see that there's a switch. This is a seizures that just happened. But after switch on the gene, so this is not the same animal because this is almost, I think a few days later it died. But from another animal where I injected to switch on the gene when the animal is already showing something similar symptoms for the left one, for the left animal, it actually can be rescued. So the phenotype becomes go away. And this gives us a very interesting results, suggesting that by restoring the gene, actually this condition is rescuable. But the next question becomes where and what's particularly what cell types that might need to be receiving this gene replacement therapy. So we know that this enzyme SSADH is expressed in the brain, but it's not everywhere and it's not expressed in the same level. So this is a molecular method understanding what is the amount of ALDH5A1 gene throughout the whole brain. So there's a mouse brain, and it's the data that's publicly available from the Allen Brain Institute and also Karolinska Institute. And as you can see here, so there are regions where you can see more of this ALDH5A1 gene expression. And if you separate by cell types, you can also see that there are some cell types that express more. So you see this like a Manhattan plot of these gene. You can see some cells that has more molecules per cell compared to the others. So that means across in different cell types in the brain, there are cells that has more expression of this ALD5A1, and there are some cells that has less. And this is also verified by another group that's showing very similar results, that now the separation of the gene or the classification of the gene of the cell type is a little bit different. So you can see that GABAergic cell, that is the neuron that has that release GABA, and many of them actually has this ALD5A1 as shown in here, these white bars. And many of these glutamatergic cells, for example, they do not have ALD5A1. So when it comes to gene therapy, we think that it will be very important to address this different cell expression profile and also their expression level. So how are we going to do that? And one way we just use the nature to tell us. This is we understand in the molecular biology that every gene is regulated by some what we call the promoter regions. So the expression of the certain gene is governed in large part by this genetic instruction, genetic genetic material that instruct the expression of certain genes. So we make use of this a promoter approach, extracting a native promoter of the ALD5A1 gene and put it into a virus. So this is a viral. This is a representation of a viral gene with a very simple genome, and that we know very well about what is the capacity, what is the size of a virus, and but we heavily genetically engineer this virus such that they are not able to replicate. So that means their viral toxicity will be much lower. And in other words, it might be quite safe for using as a carrier for a gene of interest that we want to replace in our situation. So that's exactly what we are trying to do, using a native promoter strategy and drive a gene replacement therapy. So we put these genetic instruction materials into the virus and then package into a virus and then give to the mouse. So in this case, we are not switching on or switching off the mouse, but by directly releasing this therapeutic gene payload into the animal. And this will mimic a situation in patients and in the future. But this is an animal study. So here is the growth of the animal. So this green line showing individual mice that are initially they are sick. So you see that they are underweight compared to the black dotted line, which are the average body weight of the wild type animal. And as you can see that the injection timing is the time when they are already showing the symptoms, but you can see that they are actually growing back. And a appreciable amount of the mice that are injected with this virus actually grow back to a very comparable body weight to their wild type counterparts. And you can actually see from this video how well they are doing. So one of these are actually, I didn't tell you which one, but you might be able to guess which one is actually injected with the virus. And it's living and it's way beyond the life where a otherwise not injected animal will be dying at around 21 days, but they can live over 100 days after the treatment. So this gives us a indicator of this strategy might be useful. So to summarize this part, so I talked about that SSADH deficiency is a genetic disorder, and the reason is because the deficiency lead to a failed GABA breakdown, accumulation of GABA and the GHB metabolite to a very high level, pathologic level, and in this situation, it leads to seizures. So of course, I talked when connecting with the KCC2 story, this is actually something that we are trying to understand better. Apart from the GABA receptor depletion that I talked about, maybe the KCC2 is also playing a role in the pathophysiology of SSADH deficiency. But in our mouse work, we found that the symptoms associated with SSADH is reversible. And by using a gene replacement therapy, we see a very good response from the mice, suggesting that this could be a viable strategy to treat this disorder. So I would like to, so this is towards the end of my presentation. I would like to acknowledge many mentors across my research career, starting from Dr. Stephen Moss, now at Tufts. When I first joined the lab, he moved just from UCL to UPenn, so most of my work was actually done at UPenn, and many of the lab members who has helped me across the years. And I also want to thank Dr. Alexander Rothenberg, so at the Boston Children's Hospital, and people in the lab helping me to finish this work on SSADH deficiency. And I have not talked about Dr. Spatazza, who is the director of the Intellectual and Developmental Disabilities Research Center, IDDRC, at Boston Children's. And the core facilities has been really helpful for the SSADH disorder. And also with Mustafa Sahin, we have been working on other neurodevelopmental disorders and developing therapies. And I also want to thank Dr. Philip Pearl, who is the director at the Epilepsy Division at Boston Children's, who is an expert on SSADH deficiency and people from his division. And because now we are working on towards gene therapy, and honestly, I learned a lot from Dr. Guangping Gao at UMass Medical School, who is an expert and pioneer in AAV technology. And I also want to thank his colleagues in the Gene Therapy Center for packaging the virus. So I also want to thank my funding support, including Epilepsy Foundation, in my PhD work, and now SSADH and SLC601 Connect. And to sum up, I just want to say that we are going from a very simple molecule and discover over 70 years, and to now where we know so much about how it works in the brain and how it recycled. But there are also things that I have not really talked about given the time and also the depth where I want to go. And for example, what I talked about is GABA-A receptor, but you might be curious, is there any other not GABA-A? In fact, there are GABA-B receptors. And there are a whole different topic that we probably need another day or few days to talk about. And only going back to the GABA-A receptor, we talk about synaptic GABA receptors. There are extrasynaptic receptors that it operate in a different way. And there are also chemicals or molecules that might be targeting specifically to these subfraction of the GABA receptors. And we also talk about other transporters that might be playing a role. And these are all very interesting part of the science, and that give us opportunity to understand better how GABA is regulated in the brain. And also, in many situations, these molecules might be involved in the pathologic conditions. And now we might have some tools, some knowledge that we might be able to design specific therapy for these disorders. So I'd like to thank you for your attention, and you can find my contact information here. And I'd like to thank Monica again for the invitation, and looking forward to more communication and more discussion. Thank you.
Thank you, Dr. Lee, for helping us understand the GABAergic signaling and the GABA life cycle. I have a couple questions.
Yes.
There's a question from Luke. Are there specific genetic mutations in GABA-A receptors that are more commonly associated with SSADH deficiency?
That's a very interesting question, combining the two things. We know that there are over 30 variants of SSADH, the gene mutation. How does that relate to GABA receptor is not known. So there is actually a natural history study that is ongoing in Boston Children's Hospital. What is natural history study? Basically, is understanding the development of the disease. So that will involve genetic testing and sequencing of the patients, and to understand better how these what we call the genotype is the gene mutation related to their clinical symptoms, their manifestation in the clinical setting. So this is actually an ongoing work, and we try to understand better. In terms of the mutation, GABA receptor, of course, there are a lot of flavors, if you will, of the mutation that is also known to cause disease. But that as obviously will be another topic for another day. Yeah.
Great. And then I have another question from Augustina. She wants to know, how can I know if my child's variant is synaptic or extrasynaptic?
So we go back to the GABA receptor. There are heteropentama. There are 19 subunits. So there are actually quite well-known literature. They talk about what are the composition of the synaptic receptor and what are the extrasynaptic receptors. For example, in the synaptic GABA receptors, very often than not, we can actually find the gamma-2 subunit. And versus in the extrasynaptic, we find delta subunit seems to be mostly prevalent in those population. So by looking at the subunit, you can actually guess, if you will, that this is synaptic or extrasynaptic. Now, if you want to know whether your patient is whether it's synaptic or affecting extrasynaptic, basically looking at the gene, whether the mutation is found in what subunit, then you can see whether it is affected in the synaptic or extrasynaptic. That being said, is to make things more complicated is that there are also research suggesting that, for example, in Stephen Moss' group, that there are receptors that are moving in and out of the synaptic regions. So the finite line of synaptic versus extrasynaptic is actually not that clear. And but we're talking about a biological system. Everything is very dynamic and mobile, and it's just like a chance that in the synaptic region there are more that you can find that subunit. It's not saying that they are discrete. And so you can see that the movement of this receptor moving in and out of the synaptic region is actually also one of the research directions that people try to understand better. Question of in terms of therapy, because we now also know that there might be drugs that may be targeting the synaptic versus extrasynaptic. So understanding that what fraction might be relevant for a specific condition, that becomes important. But I would say that this is still kind of like a research area that a lot to be known.
Can you give us an example of what a subunit would be?
Yeah, so they are, so for example, the alpha-1 subunit is one of the a prevalent subunit that can be found in the synaptic, and I think also some of the extrasynaptic actually. And so it's encoded by a name called GABRA1. So it's GABA receptor alpha-1 subunit. So it's GABRA1. So that's alpha-1 subunit. So if you found the mutation is found in this gene, that means alpha-1 subunit is affected. So GABRG2, for example, that will be gamma-2. So as I said, gamma-2 is one of the major subunit that is found in the synaptic region. So if you have a gene that is mutated within this gene, GABRG2, that means it is a synaptic also.
Here's the last question, and I'll open it one more time to the floor. Would you say that gene therapy replacement may offer reversible symptoms? So I guess the question would be, how do you know if giving gene replacement therapy will actually create reversibility of the symptoms?
Well, I would say this is part of the science that we need to figure out. From the presentation that I gave you related to SSADH, this particular disorder, the symptom seems to be reversible. But there are some caveat because we are looking at a animal that are still very young. And that age, when we start to treat the animal, is equivalent to a human age around in the early teens, or maybe like that maybe like third, maybe like third to sixth grade, that kind of age. So that, you know, the brain is actually rapidly developing. And so a lot of movable part, if you will, or we call it plasticity. So the brain is still very adaptable at that time. So that is also maybe contribute to the reversibility, because the the condition, yes, is already presented. The mice that we treat is already underway. They're already showing a seizure-like behavior. But we are able to reverse that because number one, they are younger. And number two, that condition seems to be reversible. That's the science that we found. Whether we can say other, a GABA-related disorder can be equivalent to this scenario, is remain to be investigated. So our hope, of course, is that these conditions are reversible, and that will be great. But we also need to be mindful about when is the treatment happen, and what might if there are some changes that is not reversible, how are we going to to make it better? So these are actually the things that we are that we can do is testable. So for example, we can make a mouse, we can make stem cells from patients, that we can also study some of these changes. And yeah, but that will be, I would say, will need to be tested case by case. It's difficult to generalize everything into into a single context.
And by case, do you mean by subunits and gain of function, loss of function?
Well, I would say yes. The best way is to really to test it discretely. But GABA receptor and the signaling might be shareable. So I think that might be some way that we can use some example to test whether this reversibility is is possible or not. It's hard to say at this level, I think.
Well, thank you so much, Dr. Henry Lee, for taking the time to help us understand GABAergic signaling and explaining work that you've done on SSADH. I think it's absolutely fascinating. We are grateful to have you part of our GABA-A community, and we wanted to thank you.
Well, thank you again. And yeah, I'm happy to be connected and looking forward to more interaction.
Great. Okay. Talk to you later. Bye.